being here to introduce a good friend of mine, Dr. Alejandro Buschmann, who comes to us from the University de los Lagos in Chile. He actually more specifically comes from EMAR, which is the Marine Laboratory down in Puerto Montt, in a little bit south of Los Lagos, where he was actually one of the founders of this facility and a director until just recently. He has also been an instructor in the Chile course that Dr. Scott Hamilton and I have been teaching on off years down in Chile. And his expertise is primarily in seaweed biology, but mostly in kelp ecology. And now in the last 15, 20 years, he's really developed a very strong expertise in aquaculture, which I think we can potentially talk about after his talk. He is one of the world's experts. And when I say this, I don't say it lightly. He is definitely one of the world's experts in the study of integrated multi-trophic aquaculture, which is taking off explosively around the world. And so Dr. Buschmann has really been involved in a lot of initiatives that have been influencing countries around the world and how they develop aquaculture in a more sustainable fashion. He is a member of the Chilean National Academy of Sciences and really helps to promote science at a national level within the country. And more selfishly for us, he comes to Moss Landing a lot. He's really been involved a lot with our work. We've been on multiple grants together. He's worked with multiple students, many of which he's sat on their committees. 10 years ago, he did a sabbatical here at Moss Landing Marine Labs. And prior to COVID, he came back often. So we really hope that once it relaxes a little bit, we're able to get him back over here. He can again begin to interact with those of you who are new to Moss Landing. He really has been an absolutely invaluable resource, and I am just really excited to introduce my friend, Dr. Alejandro Buschmann, who's going to be talking to us today about one of my favorite species in the world, giant kelp, Macrocystis pyrifera, their life history, unwrapping the ecological relevance of microscopic stages.
So can I start?
Yes, Dr. Buschmann, my apologies. You can start.
Okay. Yeah. So thank you for this invitation. It's always nice to speak with friends and young students. I participated in several PIAPIX meetings some years ago, so I know how it goes. And for me, it's always nice to develop ideas and try to induce conversations that provoke young students to move on and find new research areas like this on Macrocystis pyrifera. We think that we understand a lot of an algae like this. So many good papers, proposals, projects has been developed, mainly in California, but in different parts of the world. And but what I will try to do is showing you that we know quite few things about what all the microscopic stages can mean for completing the life history and become implicated in the type of dynamics, population dynamic that these species can present in different habitats of the world. Okay, I will start with something very easy. Laminariales and kelps are not equal. You can see in the pictures different species. I don't care which are one, but you will see some are intertidal, some are subtidal, some have an annual life cycle, others are perennial, others live in very shallow areas, others can be quite deep. So they can be present in warm, cold, temperate waters, but the amount of the region that they have colonized all over the world is amazing. It's quite large. So they offer us a very nice biological models to study population ecology. And if we move into Chilean laminariales, our diversity is much lower than the Northern Pacific, but we have some species of trabeculata. I will not talk about what I can find in some islands offshore of our coast, but in our main coast and the Chilean coastline, we can find Lesonia spicata and Lesonia berteruana in the intertidal mostly, are very similar, almost cryptical species, but genetically are different. Underwater, subtidal, we can have Lesonia trabeculata that is quite abundant and can be much more larger, and Macrocystis pyrifera, which is the same species that you have in California. Years ago, we differentiated the species because of the holdfast morphology, but right now we know that that is plasticity, morphological plasticity, and depending the type of habitat, they develop in one way or the other. In Chile, we have this the different type of morphological holdfast, the differences. So we have a quite different type of habitat in which Macrocystis can be found in Chile. From the north, and even it goes to Peru, comes to the very far south of Chile, moving into the Atlantic and South Atlantic in Argentina, and in the circumpolar islands all around the world, and the south of New Zealand and Australia, and South Africa also. So this is a species that has, and perhaps from the KELPs, one of those species that have the largest distribution around the world and have colonized very different habitats around the world. So we have Macrocystis all around our coast, and I will be speaking mostly about what happens with populations that move into these more calm waters, wave-protected inner seas in the south of Chile. And why we was interested in this, it was mostly, if you look here in these figures, protected south. They present in general a much more variable temperature, salinity fluctuations along the year. And in the north are quite stable. In the exposed souths, there is some temperature variability, but much less variation in salinity. So they live in different environments in which variation, environmental variation, is more or less important in our coast. And we have tried to understand the meaning of that, and that was what we started to do around early 2000s. And along the Chilean coast, in different places, we published here a paper describing all the variations of the populations. We found that it is reproductive all over the year, more or less like California. The difference is that we find the small belt with not very large individuals, sporophytes, as in California. Our sporophytes looks much more skinny. I will show you pictures afterwards. More, not very, with much lesser stipes coming from each holdfast. But the population dynamics is almost the same that has been described for California. But when we move it in these inner seas, which are much more calm, here is the open coast, here looks Metri, it's like a lake. It can become rougher, no, with strong winds in winter, but and has strong currents also, but the wave action is much less in these regions. And importantly, as I thought, temperature fluctuations, salinity fluctuations were changing much more than in the exposed area, and salinity moving from almost 20 per thousand up to 32, like marine waters, and temperature fluctuations between 8 degrees Celsius up to 18. And even in the past years, we have been measuring the surface waters in the canopy temperatures above 20 degrees Celsius. So sublethal, at least sublethal temperatures. So when we compare the life history of the populations that are in the exposed area with these ones that are inside these inner seas, normally in the exposed area, this is a point that is a little bit an outsider, but in general, a very stable populations. They don't change their number, their densities very much, and maintains about 4, in average, 4 individuals per square meter, per quarter of square meter. Okay, so you need to multiply that by 4. So we will have about 15, 16 sporophytes per square meter. But remember, these are not as big as the sporophytes that you see in California. But you have a compensation, you get a more higher biomass, having smaller individuals. But if we move into these seas, we start to see quite important abundances of sporophytes along the year. And these are 3 different sites. All they show it the same pattern, an increment. Remember, Southern Hemisphere, this is the summer. This is the summer that we have from late spring, an increase in the number of sporophytes, and then it's a decrease, and the numbers comes to very low in the winter. Very low numbers survive in the winters. We move into a much more detailed work afterwards, and here is a comparison between the exposed in black and the white are individual sporophytes found in the inner seas. And you will see that you can see one cohort of individuals that started in April, March. They start to get reduced numbers and they start to disappear. And you can see here is a recruitment, 1 age old individuals growing, creating a canopy, and in the next winter, you see like that. They're all dying. So you see these species have the capacity to move from a perennial life history strategy into an annual strategy. And all this capacity allows this species to colonize so many different type of habitats in coastal regions. And if you see also, this is the size of the holdfast, they start to disappear, all the holdfast, and the density of the plant also reaches 0 surviving, and a new cohort start again. And if we count the number of sporophytes, so the number of sporophytes and reproductive tissues that they have, the reproductive tissues disappear much earlier, and they start to appear later in the spring after the sporophytes have started to grow. So there are several months in which you do not find reproductive structures in the natural populations. You see, here is a surviving sporophytes. It will not survive, it will not grow again, and has no sporophylls. You see it here and here, they're not sporophylls anymore. So from a population point of view, they're already dead. So here we are showing several months, at least 4 or 5 months in which you do not have reproductive tissues in the sporophytes to seed and to repopulate the microscopic stages. In the case of the open waters, they're always reproductive. In the case in California, they're always, all year long. Perhaps you have variations during the year, but always you will find reproductive tissues, an amount of reproductive tissues at least. But in this case, it's nothing there. And all these microscopic stages must be surviving to allow the recovery of the population, or the alternative hypothesis is that they should come from other places. So Luis Enrique Estejo is a master student, and this is part of his thesis. It's not published yet. I hope that it will come to a point that we have this paper going on in the literature. So he was measuring here the density of adult sporophytes. And again, they start to disappear in June. These are 2 places, 2 subpopulations. Now they are apart about 500 meters each other. So, but in both cases, they disappeared between June and July, August, and they start to appear again October, November. So they disappeared. But the reproductive structures, tissues, they disappeared much earlier in autumn, fall. So one point, the good point to study this recruitment and the capacity of microscopic stages in this place is that we do not have other KELPs. So if I go and count small sporophytes, I know these are Macrocystis. I can not confuse them with other species. And maybe that is not the case in California, where you have all this huge diversity of species in the environment. So you can see the sporophytes in spring, how many we found, reproductive percent of reproductive tissues per square meter. They also tend to be in spring, then they increase up to in the summer, and they start to decrease in autumn. The number of spores that we can release from these tissues is nothing in spring. They are released mostly, here is 2.2 million spores per square centimeter of the fertile tissues. So it's a large number, but they start to decrease again in autumn. The number of sporophytes, you see, is mostly per sporophyte increase in summer. So if you multiply these factors, you have the maximum production of zoospores during summer and early autumn. And this is the number of released spores per sporophytes that we have been measuring in this system. And for studying how this reproductive of the spores, the zoospores colonize the substrate and regenerate the populations, we did follow in counting the number of recruits in these two places. And November, December, there was a number of small recruits, but they start to disappear. It's zero along the year. And in September, they start to appear in both sides. Before, there were none. We could not count any small Macrocystis recruits. What Luis did in this case was the density of recruits on artificial substrates. We put every 15 days substrates into the water for 15 days long, and then we brought it to the laboratory and we incubated them in the laboratory to see if they were seeded by zoospores coming from somewhere, and mostly from the population that has reproductive tissues around. No? That is the more logical source of zoospores. And they did colonize. And we brought them to the laboratory, and they started to produce during summer a large number in one case, but then there was a production of a number of sporophytes along the years. But during when we put this artificial substrata in the environment in winter, they were nothing, and they will start to appear again in the next spring. No, there was a number of zoospores. The next slide graph shows the number of recruits that we were taking from natural substrate that was seeded, and we took it to the laboratory. So it was not clean, sterilized substrates like this. It was getting what it was seeded, naturally seeded in the environment. And now we can get some months during autumn a number of sporophytes. So they were there. They were they do not appear, but they are there. So they were seeded in the summer, and if you can give them good conditions for growth, they will appear and they will be present in your samples. So they are there, they are surviving, but they are surviving a longer time in the environment somehow, and to produce a new cohort of these annual populations. This is a paper by Kinlan. Mike is a co-author also of this paper, and they have what we call an arrested culture conditions for the microscopic stages, and then after reducing the amount of light and nutrients, they provided the culture conditions, and you can see how the sporophytes were starting to grow. So they had the capacity to arrest their growth and stay in some conditions, waiting for better environmental conditions to start to grow again. And they could stay up to 2 or 4 weeks in these limiting environmental conditions. An undergrad student, Karen Gonzalez, did the same, but we push these experiments further. We maintain it in these controlled conditions up to 2 months. And then we start to see how they recover. And here is the density of gametophytes that we can have after 30 days of recovering. And you can see up to 80 gametophytes per square millimeter. But if you maintain it for 2 months, you see a reduced conditions unless you provide a good source of nitrogen and coming back. And this only in one conditions we did not find any differences. But we can have about 80 up to 80 sporophytes per square millimeter, not sporophytes, gametophytes. The amount of oogonia that these sporophytes, it's about 1/4. And in some conditions, they were producing oogonia in a much higher proportion. Here is the number of sporophytes. You can see we got sporophytes after 90 days that could be recovering in 90 days of limiting conditions in our experiments. So this data shows that we can push furthermore the results of Kinlan, and the microscopic stage, they can maintain them alive, and they can recover after long periods, still 2 months in limiting conditions, and you can trigger and making them reproductive and produce sporophytes again. So they can grow the sporophytes. I will not go into detail. In a review paper with Mike, some that we wrote, and also with Julio Vasquez, other Chilean fellow, we produced this paper, review paper on Macrocystis pyrifera, and we discussed this high plasticity ecotypes that Macrocystis can have. And we can recognize today that we have a high morphological plasticity. There is evidence of a high physiological plasticity and high life history plasticity, no annual up to perennial responses of the populations, and a high capacity now with this data for surviving under limiting conditions of the microscopic stages. So they have these capacities to deal in complex environment, and the microscopic stages, they can have also developmental strategies. And I will move into this more in detail. Okay, here is the animal populations. They colonize shallow intertidal, shallow subtidal habitats, but they use mostly secondary substrata. They live on these gastropods, Crepidula fecunda, in which you see what the largest ones are the females, the smaller ones that lives on the top of the female are the males. They revert sexes as they grow, and when they are small as males, they scrap all the small algae that colonize the female shell, and the female becomes sessile and a filter feeder. So it's a very unique system. Besides the Crepidula, which has here a small size grazer, we have Tegula, which can eat perhaps larger sizes. We have also demonstrated that. And we have sea urchins. This is the Loxechinus. It's the most common sea urchin in this area. So an experiment that we published some years ago, we demonstrated, the main idea is that we have open cages. So Tegula, Loxechinus, and sea urchins, Arbacea also, they can graze. We reduced the entrance so Tegula could enter. The small Crepidula grazers were also inside. Then we closed it. We closed completely the cages, so Tegula could not graze. Then we had only female Crepipatella. We took the males out, so the scrapping of the shells was also taken out. And then we had artificial Crepipatella or dead Crepipatella. They were not filtering, so the filter effect was taken away. And then we had naked rocks. And this was replicated 5 times in each site in 2 locations that are about 60 kilometers apart. And the results are quite nice and interesting. You see, when you get the macro grazers, and here you have the abundance, this is the abundance of the macro grazers. When you reduce the entrance in the cages, they have reduced numbers, and they maintain it at the low numbers in all the other treatments. Always some Tegulas they could enter and so on, but statistically, we controlled the abundance of grazers. And the KELP recruits, okay, remember, they were higher here and lower here. They started to recruit at increasingly higher numbers when you removed the large grazers, Tegula and the sea urchins. And, but very interestingly, when you had only the female shells without the filtering effects, Macrocystis could not colonize. And even on the rock, there were significantly lower numbers of recruits, of KELP recruits, as compared to the other treatments. So something was happening also in this that was restricting. It's not only a grazing effects, it's something else. So what other algae, a red algae, Sarcotalia, was colonizing strongly on the rocks. But on the Crepipatella, in which they had not the filtering effects, they were colonized, and up to, in one locality, up to 100% completely covered the shell by green algae. So they colonized the shell and they outcompeted the KELP recruits in these treatments. So this is more likely to show you, this is the populations and how it looks. And you see the recruits on the shells, and most likely these recruits appear in the margin of the shells, are not in the top mostly. Here you see that the male is scraping the shell, and here are their small kelps starting to grow, and they get eaten by the male Crepipatella. Here you see they were growing, other algae are growing, so very few kelp recruits can grow on this type of substrata without the filtering effects, which is quite important to get the green algal spores. In some other experiments, we demonstrated that especially green algal zoospores, which are much larger than those spores from other seaweed, are preferentially eaten, being filtered by, or more efficiently filtered by, the Crepipatella. So there are other mortality factors for kelp. This is Ali Muth, a former student from Moss Landing. She went to Chile and is coming to Chile several times. And we put in another experiment to see how sedimentation was also affecting the capacity of recruits. And clearly, when we maintain it suspended in a horizontal way, so sedimentations cannot be covering the substrata, the recruits of Macrocystis is significantly higher. So there are other mortality factors also important. Okay, so here is a kelp. This is not Macrocystis, but just to make the case, they produce a lot of zoospores. We produce male gametophytes, female gametophytes, they get fertilized, and they produce the sporophytes. That is the general knowledge. What we know now, and what I explained, is that this phase can be highly relevant to connect two time-separated cohorts of sporophytes. And if these have not the capacity to maintain themselves and at a high, density, enough that the males can fertilize the oogonia of the female, it will not be able to close the life history cycle. We know now, and especially from aquaculture systems, that the female oogonia can produce, can reproduce by parthenogenesis. And that could be one way how to close the life's history cycle without becoming fertilized. So you can, it's a system that you can have a reduction of the gametophytes, not a high number, and still you can recover the populations. But you will lose a genetic diversity. Normally, if you look the morphology of the females, males, males have thinner cells and more ramifications. The females have larger cells and produce here one oogonia. Here they have one oogonia starting to reproduce after fertilizations, to grow, to have successive mitosis, and you get sporophytes growing, get attached and start to grow. So that is the general understanding about how reproduction goes on. In this paper also with Mike some years ago, and Julio is also part of this history, we demonstrated in this short paper that these gametophytes can grow much larger, and this is a male, under limiting conditions. And one female can produce multiple oogonia, and you can have multiple sporophytes from only one gametophyte. So this is another way how you can increase the number of success of the sexual reproduction and producing multiple sporophytes from one female. And in this case, you can have possibilities that these are not consequence of the fertilization from the same male, so some genetic diversity can still exist. We have some data that in some conditions, here low temperatures, full probosole media, we could produce a larger number of oogonia per female gametophyte in these experiments. Okay, now we are understanding, we have sorry, this is in Spanish, but you will understand the names anyway, are almost the same, no? You have the sperms, they will come and fertilize the oogonia, and the oogonia will produce the zygote, and you will get a juvenile sporophyte diploid again. Okay, but these multiple eggs can become fertilized and produce these new sporophytes. But we can have several sporophytes. Are these genetically homogeneous? There is genetically different diversity or not? So these are questions. And I show you this paper was just accepted last week. So first time I presented. So the name is How the Gametophytic Developmental Strategy Affects the Growth Capacity of Macrocystis. And what we found is that we can produce different number of oogonia per female gametophyte, up to 9 oogonia per female gametophyte, and they produce a larger number of fronds. Yeah, and these are the number we were observing 133 marked female gametophytes that we individualized in Petri dishes. So when you have one oogonia, you get one sporophyte attached to the substratum. And you produce one blade or few blade sporophyte, juvenile sporophyte. But when you produce these multiple eggs, you have living very closely different sporophytes, and they grow attached together. And you see a sporophytic, what we call a sporophytic unit. And we can have these morphologies with large number of blades, small number of blades. So we have two extreme morphotypes, okay? This do not occur always when you have a high number of oogonia, because in some cases they do not get fertilized, they do not develop. And so in this case, we had 8 oogonia and the female gametophyte produced only 3 fronds. So because not every oogonia was fertilized. So when you start to, I will not show you all the data that is in the paper. There is physiological data and so on, but here is the growth rates. They grow very much when they have enough nutrients, and temperature are not very high, so not reaching sublethal conditions. They can grow very fast, almost 18% per day, very fast. And much lower when you have nitrate limitations. And the interesting thing is that when you have these multiple fronts, this could grow significantly higher than the few fronts under these more stressful nitrogen conditions. I will not go into details, we can discuss that, but the important thing is that responding to nitrogen, the nitrate reductase activity of these sporophytes, multiple sporophytes, was significantly higher somehow. The question was, they are genetically homogeneous? Not. Here we have 12 sporophytes with different number of blades. And we checked every blade by each sporophyte if they're using 10 microsatellites, if they're equally genetically equal or not. And every blade was genetically different each other, suggesting that the males that fertilize the females are coming from different males. So they're not genetically equal. And maybe, we understand that some genotypes, they can grow better. Some blades in these limiting conditions may grow better and provide us with these increasing growth rates under some limiting conditions, and especially about nitrogen. Here we have field experiments. We brought the KELPs to a KELP farm. Like here, we have marked few fronts, multiple few fronts. They grow significantly in 6 months. So this reached over 20 kilograms per sporophytes. They stay less than 5 kilograms or 6 kilograms in average. We brought KELPs also under the canopy of a KELP population, natural KELP populations, and we leave them for 45 days. And again, significantly higher growth. The weight of the sporophytes reached 40 grams and compared to 10 grams or a little bit less. Here is the initial condition. So in both cases, they grew, but much more when they had this multiple blade form. So the strategy that how they develop, how they reproduce, sexually reproduce is important. This may explain the variations that we have in the number of fronds along the Chilean coast. Central Chile, this is a more integrifolia form, so it's different, but here in the south, you see in the far south, we have these individual sporophytes that have many more types coming from each front. This is a picture of Mike several years ago in the Falkland Islands. We were counting over hundreds of types coming from one of these spores. Is this because of a strategy like we described it? We don't know, but it's a possibility, and we are moving, trying to explain these variations that we can find along the Chilean coast of the capacity of giant kelp to develop into different morphologies that produce, that could grow at different rates in the environment. So coming again to this Graham, Vasquez, and Buschmann paper, so already more than 10 years ago, we put several unknown questions that we did not knew at that time about how sexual reproduction goes on and what is the meaning of these microscopic stages. And we can tell you right now that all these microscopic stages are quite relevant for, especially in annual populations. And they can couple cohorts that are separated in time for 4 or 5 months. So they can survive, sexually reproduce in between, in this period, and grow into different morphologies that may be helpful for growing in some type of stressful environments. So we are just starting to open this black box, which are the microscopic stages of giant kelp. So for finishing, giant kelp can show complex microscopic stages developed with complex developmental patterns, long-term survival in the field. They are affected by herbivores and algal competitors, but they can find their ways how to survive. They can have a complex sexual fertilization patterns. They can produce sporophytic units that are not genetically homogeneous and present better responders at some stressful environmental conditions. These not genetically homogeneous sporophytic units showed higher yields under some field conditions. So thank you, and I hope that I could develop some questions that you can develop some questions for this data and for this information that I presented. And thank you for the invitation again. You are opening the speaker.
Yeah, Laura, this is Mike. Can I, do I guide the questions or do you guide them?
I can guide them. Just a reminder, if anyone has a question, go ahead and use the raise hand feature that's under the participants tab. And as they come in, I will unmute people.
Can I ask mine though first, Laura?
Yeah, go for it.
Okay, Dr. Buschmann, thank you very much for a very extremely well-rounded perspective of all the work you've been doing on the life history of kelp. For it looked like a couple of decades there. You kind of gave us all aspects of it. And then right in the middle, you dropped that one result by the Munoz et al. Paper where you talked about different number of eggs per female. And you showed convincing work that there's consequences, that the more eggs per female that you produce, the more sporophytes you obviously produce. And then you continued on to talk about the potential value of that to the health and physiology of the sporophyte. But I don't remember hearing any comments about why you think that those females might have been selected for to have numerous oogonia in the first place. You talked about the consequences, but I thought maybe you can give us some guidance as to what you think is the reason why in certain areas of the world you have only one oogonia per female, but in other areas like you showed, you have multiple.
We are willing to test if this cannot develop with kelps in other regions. What I'm thinking is that they may be genetically fixed in these kelps the capacity to develop these multiple eggs, and other populations do not have this capacity, and that is why they have not been described. That is the first thing. But for that, we need to go to other places, or other people must do the experiments. The second point is that there is an old paper by also a friend of you, Bernabé Santelices, which that he developed with Lawsonia during early 90s or late 80s. And he demonstrated that when you have limiting conditions, the gametophytes can grow vegetatively and not become reproductive with the sexual structures and do not develop the sexual structures. And from an aquaculture point of view, if you review the literature, there is a lot of literature that you can control sexual reproduction by limiting the quality of the light, putting out the more energy radiation like blue light, and leaving only the red light. You can control sexual reproduction. So that is also this information there. So environmental conditions, if they're limiting, if you have a small, they reduce the growth of the gametophytes. They can grow and you can create a much larger gametophyte. And these larger gametophytes has the capacity to produce more eggs per gametophyte. If you have a small gametophyte with few cells, it will produce one oogonia. But you need to develop, and that is the conditions. How do you explain it? We come along all the winter time with light limitations. not enough energy to develop and sexually reproduce. And maybe that is the phenomena that is taking place in the environment and allowing to couple cohorts of sporophytes that are separated in time for many months or several months.
Well, I really like that answer because that creates a testable hypothesis that if you go to similar parts of the world that have similar environmental conditions, you have an expectation now, or at least a prediction as to what you would expect the fecundity of the females to be. So I think that's really...
That is one of the experiments that we are willing to do.
It does look like we have a question, Dr. Buschmann, from...
Okay.
So I think it's coming from Dan Gossard, who's going to have a question for you.
Okay.
Hi, Alec.
Hi.
So for aquacultural purposes...
Sorry? You have...
Oh, sorry, can you hear me?
Yes, I can. I don't see any face, no?
No, we don't have the video on, Alejandro.
Okay.
There we go.
Okay.
Can you see me now?
No, don't worry. I hear you good, fine.
Okay, so for aquacultural purposes, it seems like using this type of chimerism might promote greater yields, at least just for Macrocystis. Do you think that, or have there been any studies examining this type of chimerism in other kelps? I think did you mention that there was one for Lawsonia? Yeah,
but the way how these chimeria develop along this idea of Bernabé Santelices is that you have individual sporophytes that are starting to grow close together, and initially they're separated, and the holdfast fuse, and they have described plasmodesmata connections between cells and in the connecting interface of both sporophytes. So there is a suggestion of physiological connections or what that could mean. But the process is that you never have a unit that from the beginning grow together. That is the difference. It starts much before as what has been described by these Lawsonia experiments earlier.
Additionally, I was wondering if you could elaborate on your speculation on I think it was, I forget, but how the individual sporophytes were processing nitrogen. You mentioned that you're willing to discuss it later. I was wondering if you can talk a little bit about that.
Well, going into the past literature, no, there is some. You need to go to these old papers of Mark and Diane Littler, another Valentinus, and what is important for productivity in seaweeds in general, no, is that the surface volume ratio. And in the paper, we showed information that the surface volume ratio of these different sporophytes is not significantly different. Even the average numbers are almost the same. So we cannot explain it because of the different morphology, but but the multiple front sporophytes, they have a higher surface. They have the same surface volume ratio, but they have a larger surface. So from one point, you can harvest more light. You can have a higher uptake per sporophyte or sporophyte unit because it's a chimera, no? You have a higher surface to take and absorb nutrients from the water, and you have the same volume, so the amount of nutrients that you are taking up is enough for, you can allocate it to the same, proportionally the same amount of tissues. So that is one feature that this multiple front has that allows you to think why they could grow much larger in nature. And the other is the genetic part. Normally, what theory tells us is that some genotypes that have the capacity to grow better, they will have increased growth, and you can detect that. So that could be also a reason. The point that we do not have, the data that we do not have, is that we could have market fronts with different genetic genotypes and see if there are some genotypes, specifically some genotypes that explain the higher growth yields. Yep, and that is something that we are also willing to do.
Ale, good. We
Yeah.
looks like we also have another question coming from another student named Sloan. So we're gonna let her unmute and ask you her question.
Okay.
Hi, can you hear me, Dr. Buschmann?
Yes.
Hi. So I read that paper a while ago that showed that in Chile you get those female gametophytes with multiple eggs, and intuitively when I read it, you know, I was really curious to see whether or not they would, you know, grow up to have multiple sporophytes and how long those sporophytes that were so close to each other could grow up together. Because I guess, you know, I would think that there might be some sort of self-thinning that might happen with individuals that are so close to each other. And I was just wondering, I guess your thoughts on that. Like how close do you think these individuals could grow right next to each other without maybe competing for light or nutrients? Yeah, I guess, yeah, that's kind of my main question.
When you start to grow these multiple front sporophytes, there is some blades that grow faster, and other at much lower rates. So they're not growing, every blade is growing equally, okay?
Yep.
So you can think about shading effects of competing for nutrients, and all those ideas maybe exist. The problem for us for calculating, for example, with red algae, they have calculated some Gini index to see where they grow, which front they grow better, and but here we do not have per sporophytes a large number of fronts or large number of blades, no?
Yeah.
So because they have 3, 4, 5, so to make statistic with those low numbers is a little bit more complicated. And the second thought that I can indicate you and try to respond to your question is that the amount of blades that these units has is not so large. We're not talking about 10, 20 together, no?
Yeah.
So even in that conditions, they're not. We do not have a good evidence about competition for light and things like that. You saw some of the graphs. In good conditions, they're growing up over 10% per day.
Mm-hmm.
So they're growing fast. So I have no indications that those are in, well, especially laboratory experiments in conditions that I'm sure that they have nothing is limiting. They have enough nutrients, enough light, good temperatures. So everything is fine for growing. But even though we have they in low numbers in our culture system, so the units do not compete each other. Mm-hmm. So, but each unit could grow at very high growth rates.
Yeah.
So
go ahead.
That's the only thing I can tell you. No, they grow fast.
That's interesting. I guess a second kind of follow up to that, in those areas where you suspect, where you kind of had photos of Mike that had individuals with a lot of stipes to them, you know, in my mind, I kind of am used to the California kelp forest. So since your forests sound like they're a little bit more sparse and thin, if these individuals that we're looking at in the lab are occurring out in the field, they also might be more spaced apart. And so, in general, when you're in a Chilean kelp forest, am I correct in thinking that, there might just in general be less light competition down there than there would be in California?
I will say our kelps, unless you go farther south, so mm-hmm. 50 degrees south at the end of South America, mm-hmm. in which you have larger kelps like in California, mm-hmm. and at much at deeper, and you find the populations in much deeper. Mm-hmm. In our case, in all the Chilean coast, open coast, the populations are rather shallow. Mm. Yeah, so they should not be light limiting. But if you go to farther South America, it's like Alaska, no, or mm-hmm. Canada. During winter, you are light limiting. The limitation mm-hmm. is there. So the strategy for growing this microscopic stage, I don't think that they are growing very fast and getting and reproducing very fast and so on. What we understand that happens in places like California, no?
Yeah, cool.
Right.
Where you have more light and better growth conditions.
That's a good stopping point. We do have one more student who has a question, Alejandro, and we're going to let Lindsay Cooper unmute and ask her question. And I think that'll probably be it for the question section. And then just allow everyone to realize we are going to have an informal happy hour after the seminar where we can take our cameras and turn them on and talk a little bit more freely and less formally. Okay, go ahead.
Hi, Alejandro.
Hi.
That was a really interesting talk. That was really cool. I was thinking that it would be interesting to see if those multiple sporophytes that came from the females that produce multiple ogonia, if they would then, if you cultured those multiple sporophytes, if they would be, if they would have female gametophytes that would also produce multiple oogonia. Do you have any interest in, or were you able to get any gametophytes from those sporophytes?
No. We were willing to have that, but we installed those sporophytes in the field, but we lose them during the winter due to a storm, and so we could not close the cycle.
I see.
So, sorry. We were not able to do that.
Yeah.
We were thinking on that idea, but yeah, the idea was perhaps selecting those that have one spore, have a few blades against those with high number of blades, and if that we can get some evidence of that trait can be have some genetic background, no, but we could not test any of those ideas.
Did you transplant all of the ones that you grew in the lab into the field?
A number, not everyone. Also, it was a little bit complicated for doing that, and we lose the transplanted individuals.
Mm-hmm.
We use some ropes that we put in the bottom, seeded with the small sporophytes, but the storm produced a lot of damage. So we had every ropes entangled together, and we could not close we could not get the reproductive sporophytes from the field.
Sorry to hear that, but we definitely understand storm damage around here. But
All right.
So not always the experiments works.
Mm-hmm.
And that's a good lesson to leave this one on. Thanks, Alejandro. Thanks, Lindsay and Sloan and Dan for also asking questions. Thanks everyone who tuned in, looking at the participant list where the participants are much more broad than just Moss Landing, and we're seeing a bunch of, I think, fans of Alejandro from around the U.S. tagging in to look at his latest presentation. So Alejandro, we want to thank you very much.
Thank you.